Our next speaker is going to talk, the title of the next speech is Influence of Anthropogenic Perturbation on Energy Trade-offs of Rock Pigeon by KJ Mathiaparanam, SS Seneviratne and PV Udagama.
Good afternoon, ladies and gentlemen. I am Mathiaparanam from the University of Columba, and I am here today to present to you some of the findings of my undergraduate research project, which concluded earlier this year, titled Influence of Anthropogenic Perturbation on Energy Trade-offs of the Rock Pigeon, Columba, Livia. As we all know, every organism has a finite energy budget from which various amounts of energy are allocated for different life history processes to attain an optimum level of fitness. When animals are infected with diseases or parasites, the stress on the immune system increases, thereby causing a higher allocation of energy for immunity at the cost of the other life history processes, to cause a deterioration in the fitness of the organism. We also know that different types of organisms deal with similar infections differently owing to various reasons. Similarly, similar types of organisms of the same species or the same type, also may deal with similar infections differently, owing minutely to genetic variations and more largely due to environmental variations. In this study, we tested whether anthropogenic perturbation caused by urbanization acts as an external stressor to influence this energy trade-off system, to cause a deterioration in the fitness of organisms. Looking at the methodology, we were dealing with different levels of anthropogenic perturbation, so we had to work with a model species that could be found under different levels of anthropogenic perturbation. So we chose the rock pigeon, Columba livia, because in the captive setting it is like a fowl, in the urban setting it is like a crow, and in the wild setting it is like any forest bird. So we had populations from different levels of urbanization or anthropogenic perturbation. Our least perturbed population was from the Pigeon Island National Park, which was the only wild rock pigeon population in the region. Then we had a semi-wild population from the mainland of Trincomalee, two urban populations, one from where I come, Columba University, and then another from Caledonia, with considerable levels of urbanization or anthropogenic perturbation, a partially captive population where the pigeons were allowed to fly and return to their cages by dusk, and also the highest level of anthropogenic perturbation which was seen in the completely captive population where the pigeons were held captive 24/7. Upon capture, we weighed the pigeons, after which we took some morphometric measurements, the head length, the beak length, the length of the tarsus, and after that we assessed the cutaneous fat deposition of the abdominal region visually and also by feeling the keel area, and assigned a fat score from 1 to 5, where 1 was assigned to the lowest fat deposition and 5 to the highest. We also collected blood from the medial tarsal vein, and this blood was used for hematological and immunological parameters and also for screening of hemoparasites. We used a sandwich ELISA method to quantify the IL-6 cytokine levels, and SDS page was used for total immunoglobulin quantification. We screened each pigeon for ectoparasites, and these parasites were collected and preserved in 70% alcohol until further identification. We looked at 4 different parasite load indices: ectoparasite load index for ectoparasites, parasitemia for hemoparasites, prevalence and incidence for both hemoparasites and ectoparasites. We had 5 immune response parameters: the WBC count, the differential count, the platelet count, IL-6 cytokine level, and total immunoglobulin content. These 5 parameters were converted into a score from 1 to 5 to standardize them using that formula, and these scores were averaged to calculate an overall immune response score. Similarly, for the physical fitness parameters, fat score, body condition index, pigmentation score, RBC count, PCV and MCV, we converted the parameters into a score from 1 to 5 to standardize them using the shown formula, and these scores were averaged to calculate an overall physical fitness score. These three calculated indices were then statistically analyzed for significant variations among the 6 populations using one-way ANOVA, and significant correlations between the 3 indices were tested for using the Pearson's correlation. Looking at the results for parasite load, we observed 4 genera of ectoparasites, Columbicola columbae, a Campanulotus species, a Colpocephalum species, and Pseudolincia canariensis, or better known as the pigeon fly. Both the pigeon fly and Columbicola columbae, shown in the first and fourth columns, did not show a significant variation in terms of prevalence among the 6 populations. However, the Campanulotus species was found in very low prevalences in the wild and urban populations, but was not found in the captive population. On the contrary, the Colpocephalum species was not found in the wild population, but was found in the urban and captive populations. And in the captive populations, they showed a very high prevalence, which led to a significant increase in the ectoparasite load from wild to urban to captive populations with an increasing anthropogenic perturbation. We observed one species of hemoparasite, which was identified as a Hemoproteus species. However, the parasitemia of this species did not show a significant variation among the 6 populations. Looking at the immune response parameters, the WBC count showed a significant increase from wild to captive populations. So did the heterophil percentage. However, the lymphocyte percentage and the platelet count both showed a significant decline, while the IL-6 cytokine level and the total immunoglobulin content both showed a significant increase from wild to captive populations, collectively contributing towards a significant increase in the overall immune response score from wild to urban to captive populations. Looking at physical fitness, the FAT score and the BCI score, or the body condition index, both showed a decline from wild to captive. The pigmentation score did not show significant variation among the 6 populations. The RBC count, the PCV, and the MCV, all 3 showed a significant decline again from wild to captive, thereby contributing to a significant decline or deterioration in the physical fitness score from wild to urban to captive populations. Looking at relationships between the 3 indices, we observed a significant strong positive correlation between the ectoparasite load and the immune response score, significant negative strong correlation with between the ectoparasite load and the physical fitness score, and again another negative, a significant negative correlation between the immune response score and the physical fitness score. So the final conclusion of this study in a nutshell is that with increasing anthropogenic perturbation from wild to urban to captive populations, the ectoparasite load increases significantly, thereby causing a stress on the immune system, which inevitably causes an exacerbation in the immune response, which in turn causes a significant deterioration in the physical fitness of organisms, most probably through a shift in the energy trade-off system that I explained to you before. That's about it. I would like to acknowledge the various institutions and individuals who supported this project, and these are my references, and thank you for being a wonderful audience.
Thanks for keeping you to your time. Thanks. Firstly, my apologies for not pronouncing your name properly. Can you tell me how it is pronounced?
It's Mathiaparanam.
Mathiaparanam. Mathiaparanam. Okay, you stuck to your time, but the previous speaker took another 3 minutes, so you don't get any questions. Oh, sorry. Any questions? So, no, I was just joking. Any questions? Sorry. No questions? Okay, I'll make a comment then if there's no questions. I think I made it a point when I started that our research needs to be applied driven. But having said that, I say these things because I'm mostly commercially driven. My background is commercial development. But it is also necessary to have these studies because the end result of these studies is to change socially. I think we advance as a society by learning all these aspects of our own lives. So the research that you've done in this space is not of low value, but it contributes enormously to the society. So there is a massive value in doing this sort of research as well.
And I would like to talk about the application of the study actually.
Yeah.
We are commercial oriented, but for the survival of humans we also need animals and trees.
Absolutely.
So we look at conservation, but the outcome of these researchers just question whether we are really conserving, because we have national parks and protected areas.
Absolutely.
But there are human beings everywhere.
Absolutely.
In the middle of a national park we see PVC tubes, so that means there is human intervention.
Yeah.
So with that level of human intervention, it is going to cause an adverse impact on animals and plants, and in turn to humans. So we need to look at those aspects as well to go forward.
Absolutely. There are a lot of indirect benefits to the society. It's not only money earning exercise. So I think it's a very well done research project. Thank you.
Thank you.